start?
Yeah, whenever you're ready.
Okay, so we're Gut Feeling. I'm Haley, this is Afifa, and this is Olivia, as hopefully you know by now. So our mission was to improve on current gut-on-a-chip technologies to try to empower breakthroughs in intestinal and microbiome research. So what is a gut-on-a-chip? Well, it's a microfluidic device that is designed to mimic the structure and functions of a human gut. And why use a gut-on-a-chip? Well, up to 90% of diseases can be linked to gut and microbiome health. And so the gut-on-a-chip model enables us to isolate the gut system from the whole organism. This facilitates research, enables more control and more dynamicism in studies. The gut-on-a-chip can also enable one to simulate a individual gut environment, which enables personalized drug treatment optimization. And then the gut-on-a-chip also provides a better alternative to other models such as animal models in that it's more accurate and reliable and humane. So current gut-on-a-chip models largely use PDMS, which is frowned upon by for drug study purposes because of its absorbance properties. The current chips also tend to lack the more realistic representation of the 3D architecture in the gut, so villi, as well as flow. And current models tend to be very difficult to fabricate, complex processes that are also hard to then mass produce. This is why they don't gain widespread adoption in pharma and R&D. Also because big pharma tends to use robotic pipetters and multi-channel pipettes and whatnot, which is not compatible with these chips. So our objectives were to use a material other than PDMS because of that absorbance issue I mentioned, as well as to create a fabrication process that was very easy to replicate, and to create a physiologically accurate villi model within the chip and successfully seed CACO2s within the device. So, CADing and fabrication is our first step. Our materials we used were PMMA, which is acrylic, also the secure seal adhesive sheets, and PEEP membrane. These were chosen because they're biocompatible. They're translucent, so it's easy to image our cells. They're also cost effective, relatively inexpensive, and they lack absorbance, as the PDMS has. First step was to laser cut. So we took the adhesive sheets, stuck that to our acrylic, and then laser cut the geometry shown here. These are our 3 layers. Then we assemble the device. So we take off the protective covering on the adhesive sheets and stack the layers together. Then we use a clamp and hold that for 5 minutes to create a tight seal in the device. Then we set the device in a vacuum chamber and let it degas overnight. This helps to remove any remaining bubbles between the layers. And so this shows an evolution of our device. The first, over there, our original device, we had that large wide channel. We found this is not practical. It uses more media than we need to. Also, it had leakage problems. So then we moved on to our second iteration of the device where we shrink the width of that channel. We also started using the clamping strategy to create that tighter seal and avoid leakage. And then we have our third iteration of the device. We changed the hole dimensions there so that we could interface with the tubing that connects to the pumps so that we could integrate flow in our device. And to test for leakage, we injected food coloring in water or media. And the third image you can see here is successfully passed the leakage test, as this was taken after 24 hours in an incubator with media injected, and there was no leaking.
So next I'll talk a little bit about our hydrogel layer. So just to give you a little bit of background, there's a lot of work right now in the scientific community where they're looking at scaffolds and they're showing that having 3D architecture actually guides cell proliferation and differentiation. So this is a paper from Shahab et al., but what you can see here is there's a normal column and then you have cells that are growing without a scaffold and with a scaffold. And as you can see in these pictures, when there's a scaffold, you see that the cells are differentiating and they're taking on the form that they should be. So this was kind of our motivation for including this hydrogel because, as you guys may know, The intestines involve both the villi and the crypt, and those structures are very important for the differentiation of the many different types of cells that are in the intestine. And to kind of do that, we decided why not take a hydrogel, create villi structures, and then incorporate it into our device. And so we had a few aims with that. First, we wanted to model anatomically accurate villi and crypts. Then we wanted to be able to fabricate a hydrogel layer with the villi and crypts. And then lastly, introduce that hydrogel layer into our microfluidic device. And so ideally, once we create our hydrogel layer, it'd be sitting right here on top of the membrane. So that'll be in the channel where the cells are. And so the hydrogel material we were using was PEG-VS. We haven't been able to actually start working with this, but we've been talking with Katie Lam from the Griffith Lab, and she was very kind to provide us with this hydrogel. And it's hydrophilic, it's biocompatible, and it's widely used in a lot of tissue engineering and scaffolds. So it holds a lot of potential. So the first thing we did was we catted our crypts and villi. This is stuff that Olivia's been working on. But on the left, you can see that the stuff that's protruding are the villi, and then the little indentations are crypts. And then, obviously, to create this, we would have to create a mold, and this is where our negative is. And the process of creating this, we did a little bit of research, and we came across this paper that uses calcium alginate molds. And the nice thing here is that calcium alginate is a material that dissolves in EDTA. And so rather than just having a plastic mold, you can create a mold out of calcium alginate and then cast your hydrogel. And instead of using just pressure to remove the hydrogel or the process potentially damaging your structure, you can just dissolve the calcium alginate and keep the structure that you want. And so our workflow was taken from this paper by John Hwan-Sung et al. And so the first step is to 3D print a plastic mold and then create a PDMS villi structure, which is the mold for your calcium alginate. And then you create your calcium alginate mold and then cast your hydrogel and hopefully get your villi and crypts. And so taking this forward, we've been kind of trying to see if this is just a viable protocol. And to do that, we kind of let go of the crypts and the villi, and we're trying to just create a basic hydrogel rectangle using this method to see if it works. Because if it works, then it's very easy to just find a printer that can create your crypts and villi and then use the same process. So far we've been able to 3D print our mold, we've been able to do the PDMS casting, and we've been having some issues with acquiring calcium alginate. So this is something that we're hoping to get done next week, is creating the calcium alginate mold and then the PEG hydrogel. But we're really excited to see if this will happen next week, because I think it's really exciting to see if we can incorporate this 3D structure, and I think it'll add a new element to our design. And Olivia will talk about how we see it ourselves.
So now that we have our fabricated device, we need to add the cells. So first we chose Keiko-2, because there have been a lot of studies published using Keiko-2 cells, so we could sort of look back and see which methods worked well or which didn't. And then Keiko-2 spontaneously differentiates into a monolayer with many of the properties of typical enterocytes found in the intestines. And on top of that, the tier values measured from Keiko-2 more closely model in vivo values than other monolayers such as HT29. And Keiko-2 also expresses drug metabolizing enzymes, which is good for studying things such as drug absorption. So to seed our cells on the PEAT membrane, first we prepared our devices by running ethanol through the channels and then aspirating and then doing the same with water. And then to prepare our cells, we started by splitting, and then once we spun them down and got our cell pellet, we diluted it with media and then counted. And then once we determined the amount of cells and the volume to add to our channel, we added media into the bottom channel of our device and then cells into the top channel. And each device contains about 100,000 cells. So as you may have seen a few slides back, there was a photo of our cells that we actually used to seed in our device. But they were about 90% confluent, so when we added them to our device, they no longer grew, and so this is what you see now. So basically our device demonstrates that PMMA is a viable alternative to PDMS. And we also created an economically feasible device, as none of our materials are super expensive, and our fabrication process is relatively easily replicable. And we also explored the potential for the villi integration, and we demonstrated a potential to seed the Keiko-2 on the cells, but so we will attempt to do that again this weekend or this coming week. So looking back at the semester, we would have made a few changes to our approach. from the outset, defining goals instead of spending a lot of time going back and forth and debating which needs we wanted our device to fulfill. And then I think improving communication would have been better to set deliverables and set meeting times for each week would have helped us stay on schedule. And then as well as designating a leader from the beginning so that we all know if there's an issue who to go to, who has a good idea of what's going on over all parts of the fabrication process. And over the semester, we've adapted to changes in team composition. So our team did shrink over the semester, so we had to sort of redistribute roles. And then we've definitely learned about how to communicate and present clearly on scientific research and findings. And then I think since we all came from very different backgrounds, some of us didn't have experience with cell culturing or CADing, so we all learned a little bit of that. So takeaways from the gut-on-a-chip field. There's always room to build on what's already been done. So while the gut-on-a-chip field has been continuously evolving, there's still always going to be room to add to better that. And then there are people constantly creating novel ideas for guts on a chip, but they don't always cater to commercial needs because commercially they usually use 96 well plates as well as an automated loading system, and many new guts on the chip aren't compatible with that. And then the gut on the chip only models a very small part of the human body, a very small system, and so it just goes to emphasize the complexity of our bodies. And then to create a functioning microfluidic device, we really need a lot of interdisciplinary cooperation too. So these are images we took of our initial 3D printed mold to create the hydrogel villi. So as you can see, the one on the left is kind of blurry, but it shows something that's may be sticking out or protruding from our 3D print, and so we can't tell if that's a crypt or not. It's pretty inconclusive. And then the image on the right shows either a protrusion or indentation that appeared at several points along the channel, but the dimensions aren't correct for either our villi or the crypts. So chances are that the mold did not print correctly, which is why moving forward, we'd like to accurately create the initial hydrogel stand, which can be done either by sending our CAD to an outside company who has a printer with a resolution small enough to print our CAD, or we can work with Pierre in the Griffith Lab, who has a printer that would be able to print our device, but unfortunately it's not functioning right now, which is why we weren't able to try it out. And then we can further explore laser ablation because we tested the laser here and it has a resolution of about 0.6 millimeters, but we really need something around 0.17 millimeters to accurately model the villi. And once we have created this hydrogel, we would like to be able to incorporate flow into our design. And then once that's established, co-culture. And then once our design is functioning, we'd like to be able to scale it up to be compatible with multi-channel pipettes. And we'd just like to thank Timothy and Rebecca for their help this semester, as well as Katie and Dr. Griffith and Dr. Alm. And then shout out to BioJax for helping us out.